And today I have a pleasure of introduce you Dr. Alberto Vázquez-Salazar. Alberto, I met Alberto 12 years ago, still an undergraduate student, I think so. And during this time, Alberto became a very important young research on the origin of life. It's a big pleasure to me have you here with us today. Alberto, thank you very much for accepting my invitation. And please go ahead.
Thank you very much. Good morning, good afternoon, good night for everyone around the world. And thank you, Savio, for the kind introduction. When Savio asked me if I wanted to give a talk, I couldn't say no, because we have known each other for quite some time now. We've known each other since I was a very young undergrad student in Antonio Lascano's lab at UNAM in Mexico City. And now I am a NASA postdoctoral fellow working in Irene Chen's lab at UCLA. And I am very, very happy to be here to share with you some of the results we've had working with catalytic RNA molecules, working with ribozymes, and also just to tell you about how awesome and how amazing it is to have this potential of mimicking the Darwinian evolution in the test tube. Okay, I think there was some microphone must be still on for the audience. But if everyone can mute the microphone, we can continue. Okay, so as Savio mentioned earlier, this year we're commemorating the 100th anniversary of the publication of the book, The Origin of Life by Alexander Oparin. And what Oparin did was set the basis for the formal study of the origin of life. And we just have, at UNAM in Mexico, we just have a couple of panels and conferences commemorating this. So it seemed very appropriate to start my talk by mentioning Oparin's work. And as I said, in 1924, the theory, Oparin's proposals was published, and it involves a reducing atmosphere and a synthesis and accumulation of organic compounds, which resulted in these gels or micro droplets that were rich in these organic compounds. By 1936, Oparin will adopt the concept of coacervates, from which the first life forms are proposed, or he proposed, evolved. And these first life forms for Oparin will be something similar to anaerobic heterotrophic bacteria. Now, today, the heterotrophic origin proposal is still very valid, and in my opinion, is the most scientifically robust proposal about how life could have originated. In fact, it has adapted and it has absorbed some of the most important discoveries in molecular biology and biochemistry in the last century, including the discovery of catalytic RNA, the discovery of ribozymes. This discovery, the discovery of ribozymes, gave a lot of strength, a lot of strength to a proposal known as the RNA world. And this RNA world, many of us are convinced that is the evolutionary predecessor, the ancestor of contemporary life. So this is a very classic scheme in the field of the origin and early evolution of life. And in here we can see all the events that led to the that led to the origin of life, and then the main events during, during the very early evolution of life. And today, for this part of the talk, I'm going to focus on the RNA world. But what is the RNA world? Well, the most compact or classic definition of the RNA world says that it's a stage in the early evolution of life where the catalysis of chemical reactions was given by ribozymes, by catalytic RNA. And this perpetuation, the perpetuation of these catalytic ribozymes was given by the cell replication of those same RNA molecules. In this case, in this sense, the RNA world was replicative RNA and catalytic RNA in a cycle. And this was enough to sustain the early stages of life. But as I said earlier, this is a very compact definition, since in reality, the RNA world means different things to different people. And why is that the case? Well, because the RNA world hypothesis is a group of related models. And as models, they have a variety of assumptions and definitions. However, all the hypotheses, all the RNA world hypotheses have the following points. Number one, at some point in the evolution of life, the genetic continuity was ensured by RNA replication. Number two, the Watson and Crick base pairing was the key to such replication. And number three, genetically encoded proteins were not involved in catalysis. And with these three basic assumptions, we can explain and explore most of the different versions of the RNA world. So let's, for instance, start by analyzing an example of how contemporary metabolism may have evolved from this RNA world. Here we can see how a ribozyme in the RNA world can start to interact first with amino acids, but also with peptides. And these peptides could have conferred structural stability, or maybe even a greater catalog of chemical groups for catalysis. And as the time passes, and once the protein synthesis machinery originates and evolves, these peptides can grow. And as evolution passes, these RNA protein complexes could have two fates. One, they can establish themselves as increasingly larger and larger RNA protein complexes, such as the ribosome that we have in today's cells, or they could have evolved into protein enzymes, where the original structure of the ribozyme was completely replaced by protein structure. So if you notice here, these two types of catalysts, these two types of molecules are precisely what we find in the cell today. Although, of course, the dominant catalysts are protein enzymes. So what evidence do we actually have for this scenario, for an evolution from the RNA world? Well, the catalytic and structural properties of RNA, as well as their extraordinary ubiquity in cellular processes. For instance, we have here a RNA molecule, and we have large non-coding RNA that, for instance, can go and be part of the ribosome. We have small non-coding RNA, such as tRNAs or other small non-coding RNAs. We have coding RNAs, such as the messenger RNAs, that sometimes include very complex structures in the form of riboswitches. We even have modified nucleosides that have a very specific function in their own, such as ribonucleotide coenzyme, alarmones, antibiotics, or other cases of very highly modified ribonucleotides or molecules that cross paths with ribonucleotides, such as histidine. And all of this is consistent with the proposal that this molecule, RNA, play a much more conspicuous role during the early evolution of life. And what we have here, it's kind of an updated figure with the same information, but I wanted to present the figure because this was recently submitted to the Genetics and Molecular Biology Journal, which the invitation to submit a manuscript to that journal originally came from Sabio also. So in that manuscript, we review the literature, and we also discuss the roles that RNA catalysis played in early genetics and in the early evolution of life. And in that paper, we discussed mainly three things: how catalysis of RNA was very important, specifically because of transesterification reactions, that this type of reactions make RNA extremely reactive, sometimes at a point of being self-cleavage, and that's something that we really sometimes don't want, but sometimes it's also beneficial. And the other thing is obviously genetics and how the RNA world, a protocell in the RNA world, would have had a genome made of RNA in an analogous way, such as RNA viruses have today. I am not saying that viruses come from the RNA world, but it is very interesting that viruses have RNA genomes, and this feature makes them good candidates to study the dynamics of an RNA genome in the lab today. But again, I am not saying that the viruses come from an RNA world, just that they are good models to study RNA genome dynamics. So again, these are two very important roles played by RNA in early genetic system: catalysis and genetics. And of course, we have also here a self-replicating ribosome, because this is like the hypothesis of how RNA ribosome could have self-sustained in a very early stage. But beyond those very specific examples for catalysis and very specific examples for genetics, in the early RNA world, ribosemic activities may have played a huge role for the continuity and for the homeostasis of these early protocells. For instance, we have activities such as self-modification, in which the RNA molecule interacts with a substrate and incorporates that substrate into their chemical structure. We have terminal and internal modifications. We have phosphorylation in a system that is perhaps analogous to what happens with ATP today. And of course, we have polymerization, that it's very important because it's allowing us to replicate our RNA genome, our ribozymes, and also in the polymerization process, just as it happens today, we can introduce mutations, we can introduce errors, and that is the field for evolution. So I would love to talk about all the examples that are reported in the literature today about all these activities that are very important for an RNA world. But of course, because of time, I can't. So I'm just putting here some of the references that are there about all the efforts around the world that labs have been carrying out to discover new activities for ribozymes that may have been important for the RNA world. But not only that, there's many other labs in the world that are working with RNA, but not necessarily to look into early evolution of life or to look into the RNA world, but to actually implement RNA technology in therapy and in other applications. And I'm just putting this example of a book that I found on the internet that summarizes a lot of these examples of how people can actually work with RNA and ribozymes and other functional RNA molecules, such as riboswitches and many other functional RNAs, not necessarily to investigate early life, but also to have a biotechnological application. So all of these results, all of these papers, all of these new molecules that are being discovered, it's an amazing field. And how can we actually go to the lab and start to look for new or hidden activities in our RNA molecules? Well, for that, we have CELEX. CELEX, which is systematic evolution of ligand by exponential enrichment, but you can also find it in the literature as in vitro evolution or in vitro selection, depending on who you ask to. Celix is a very simple, but also very powerful tool that we have in order to discover new functions for RNA molecules. And here I'm just going to briefly summarize a very general approach. And this is the most general scheme we can have for Celix. There's many variations based on the desired activities that we want to select. But the main skeleton, the main backbone of the Celix is this. We have to start our experiment with a library of RNA. This RNA has to have some sort of diversity in the sequence. So sometimes there's one or two, but usually it's with one random region of RNA within the sequence with randomized nucleotides. So we have this pool of RNA, and we incubate it with a target molecule. Perhaps it's a molecule that we wanted to bind to the RNA. So we incubate our RNA library with the target molecule, and we let it react for X amount of time. And after that time passes, we have to have also the ability to separate the RNA molecules that actually bound to the target molecule from the unbound molecules. So once we have the bound sequences, the bound RNA molecules, we can amplify the sequences by RT-qPCR and then generate a new library. That library is going to go through another cycle of Celix, and we can repeat this cycle as many times as possible. Again, this is just a very general scheme of how the experiment works. There's a lot of variations. It will depend on the specific needs of each person, each researcher, and each lab. But this is basically how we do Celix. So as you can see, very simple, but extremely powerful. And I wanted to show this specific example of how using Celix, researchers can select for polymerase ribozymes, starting with a molecule that has this activity, is a class 1 ligase. Several cycles of Celix. After there's still a couple of microphones open, so if we can mute them, that'll be amazing. But okay, so starting with the class 1 ligase and using Celix, these researchers were able to select and to discover these polymerase ribozymes, and you can see here in the selected ribozymes, you can see by color it matches the embedded structure of the original ligase. But now these ribozymes are primer template, or this one uses a highly repetitive template sequences. And not only that, because of all the technology advancements, all the new methodologies that are being used in the lab today, some of the things we can do once we have our selected ribozymes, it's, for instance, try to see if we can solve the three-dimensional structure. So this is a paper that was published a couple of months ago, I think February this year, and they worked with an RNA polymerase ribozyme that was also selected using in vitro experiments, Celix. And what they did was use cryo-EM to solve the three-dimensional structure of the polymerase. So in this case, we are seeing that the polymerase, this specific example, is a heterodimer consisting of a catalytic subunit, which is here in orange, and a scaffolding subunit here in cyan. And what we can see here is the three-dimensional structure of that ribozyme. So thanks to advancements in methodologies and thanks to the implementation of cryo-EM, in this case, we can see at a very good resolution, 5 angstroms of global resolution, we can see how our engineered or in vitro selected ribozymes look like. And of course, with all these molecular information, atomic almost information, we can see the actual, or propose at least an actual mechanism for the reaction of the ribozymes and how they interact, in this case, with each monomer, etc. So if we couple all of the techniques available and all the new advancements in methodologies, we can have very interesting results. And this one, specifically, it is relevant for early evolution of life because the ribozyme that they are studying is an RNA polymerized ribozyme, one of these ribozymes that would have been polymerizing RNA in the very early stages of evolution. But let's go back to this table, to this scheme of the several ribozymic activities that may have been important in the RNA world. And now I'm going to focus on the self-aminoacylation, self-modification type of activity, specifically in self-aminoacylation ribozymes. That means that it's going to be a ribozyme that is going to incorporate an amino acid into its chemical structure. Why aminoacylation? Well, aminoacylation is a very important reaction in the modern cell. And this is a scheme that illustrates how aminoacylation happens in the current metabolism. So aminoacylation of the tRNA is carried out by this huge enzyme that's called aminoacyl tRNA synthetases, or AARS. So as you can see, this AARS charge the specific amino acid into the proper tRNA. And once the tRNA is charged with the amino acid, it can be incorporated into the ribosome, and then the protein synthesis can continue. But what happens if we think about this process with an evolutionary perspective? We can hypothesize that ribozymes will have self-aminoacylated using activated amino acids to enable the synthesis of peptides and then proteins, acting both as tRNAs and the aminoacyl tRNA synthetases at the same time. In this way, simplifying this scheme a lot. So with this idea in mind, and with this hypothesis, evolutionary hypothesis in mind, in the lab of Professor Irene Chen at UCLA, one of the goals a couple of projects ago, like 5 or 6 years ago, was to select self-aminoacylating ribozymes using CELEX, using in vitro selection. So this is the specific CELEX approach that they used in the lab. I'm going to explain it very briefly. It starts with a DNA pool. This DNA pool, these blue or red boxes that you can see represent a 21 nucleotide random region in the sequence. And what's very important here is that this random region, the DNA pool containing this random region, represents all the possible sequences for those 21 sites. So in the experiments, they had at the very beginning 4 to the 21 possible sequences. And this means that when they started the experiments in the DNA pool, they had 99.9999% of complete of sequence coverage, meaning that with those sequences, with those molecules, all or almost all, but virtually all the complete sequence space was covered. So this DNA pool is transcribed into RNA, and this RNA is now incubated with a substrate, which in this case is biophenyl methyl tyrosine oxazolone. This substrate is an analog of the amino acid tyrosine. You can see here the methyl tyrosine part. This is the oxazolone part. and this is the biotin part. And biotin is important because when some of these molecules in our library react with BYO and self-aminoacylate, we're going to be able to capture those sequences using the streptavidin. Streptavidin is going to bind to biotin, and that's how we can pull the sequences to try to do RT-PCR, go to DNA, and then transcribe into the RNA pool again. So as I said before, for a CellX experiment, you have to do these cycles several times until you start to see the enrichment. And for this experiment, they saw that enrichment at round 6. This means that after 6 cycles, after 6 rounds, the enrichment of self-aminoacylate and ribozymes was observed. And what is more important here, or equally important, is that all the sequences that were found at this round can be sequenced, and thank you to methodologies developed in the lab, such as kinetic sequences, we can determine the catalytic activity of every single sequence. The catalytic activity of a ribozyme, we are equating that term to the fitness of the ribozyme. And with all this information, with all the fitness values, all the catalytic activity values for all the ribozymes that were found in this round of a selection that started with a completely coverage of the sequence space, what we can do is map the comprehensive fitness landscape for these 21 nucleotides. The fitness landscape for these self-aminoacylating ribozymes looks like this. And as we can see, there's clusters or families of related sequences with their associated catalytic activity, which again, in this case, is the fitness. And what we can see here is that there are paths that connect one ribozyme with another. Every single circle represents one ribozyme. And this straight line here means that there's one mutation to go from A1.1 to this other ribozyme. This dotted line represents several mutations. And this is an interesting example because that means that we can go from a ribozyme that has a good catalytic activity, it's got good fitness, but after several mutations, the catalytic activity of the ribozyme is lost. And as you can see, this cluster of sequences is actually disconnected from the rest of the families by these valleys of low fitness. This characteristic make this fitness landscape a frustrated fitness landscape, in which after several mutations, random mutations that is, the catalytic activity of the ribozymes can be lost. And to reach the next peak of activity, natural selection, or in this case, artificial evolution, should have to jump from this mutation to this one. But to explore the whole fitness landscape, it will be mutation pairwise, it will be frustrated. So I'm going to talk about a little bit of what happens during the in vitro selection to create this type of fitness landscapes. And the reason is that in vitro selection establishes a threshold activity. And this threshold activity or C level, which we represent in gray here, what it does, it eliminates sequences of low fitness. And in this case, only features of the fitness landscapes above this threshold can be experimentally determined. In other words, the vast majority of the sequences in the sequence space are going to lack activity. And this is dependent on the environment. So when we start the in vitro selection, we're just going to see the very best catalysts, the very best ribozymes. So at the end of the analysis, what we're going to have is just the winners. Well, yeah, a lot of the sequences that start the experiment are going to be lost basically since round 1. And something that is very interesting and perhaps very logic to understand is that the fitness landscape changes in the environment. What does this mean? This means that given a phenotypic space, if we change variables such as the pH, temperature, interaction with other molecules, or ion concentration, for the same genotypic space, our fitness landscape is going to look completely different because the effect of all these elements are, the effect is huge on how the fitness of the ribosome is expressed. For a very concrete example, we have the effect of crowded environment. And as you can see here, we have a disconnected fitness landscape, but the landscape becomes near neutral as we approach a crowded environment. So the environment is extremely important for defining the fitness landscape of our ribosomes. But let's go back to our original experimental results. Let's go back to our original self-aminoacylating fitness, self-aminoacylating ribosomes fitness landscape, because I want to talk about specifics of the ribosomes that were discovered in this experiment. So for instance, I'm showing in this slide 4 of the ribosomes that were discovered. All of them have these names: S1A, S1B, S31, or S21. And we have, for the past couple of years, we have characterized them. And we know now that we know a lot of things about them. One of the things, for instance, where the active site is. The active site is always located in an internal nucleotide in the sequence of the ribosomes. That means, which is here pointed as the red arrow. This means that the substrate, our analog amino acid analog, is going to bind to these specific nucleotides and only those nucleotides. We did the experiment several times and we realized that this is an actual active site. It's not random how the substrate is binding to the ribosome. It's always to the same nucleotide. What other things have we discovered with these in vitro selected ribosomes? Well, there's a substrate promiscuity between them. Here I'm showing 2 ribosomes, S1A and S2A. And what we did in this series of experiments was to test the active site to see if they could perhaps interact with other side chains. So we proved phenylalanine, leucine, valine, isoleucine, methionine, and tryptophan side chains in the oxazolone form, all of them hydrophobic amino acids. And it turns out that the ribosomes can interact with those substrates, can incorporate them, can be self-aminoacylated using those substrates. And the preference for each substrate varies within the ribosomes. For instance, S1A prefers phenylalanine and tryptophan, so the cyclic side chains, whereas S2A prefers, has no preference and can interact and can react with basically all of them. So when we tested many more ribosomes, thousands of ribosomes, we found that the specificity of a ribosome increases as the overall activity increases. What does this mean? That a high promiscuous ribosome, so a ribosome that can interact with several substrates, is going to have less activity when we measure the catalytic rate of that ribosome. But a ribosome that is more specific or less promiscuous is going to have a higher catalytic rate. And this is extremely interesting when thinking, for instance, about how new substrates, new amino acids were incorporated into the genetic code. If we think, for instance, that the potential for ribozymes with activity for a selected substrate to adapt to other amino acid substrates can be beneficial for expanding metabolic chemical space and incorporating new compounds into increasingly complex systems. And in talking about the genetic code, this will mean that self-amino acylating RNAs that react with the early type of amino acids will have promiscuous activity on the late comers, allowing co-option of these ribozymes to incorporate a new substrate once they become available. This means that if a system starts having ribozymes that interact with only the available amino acids, once the new or late amino acids come into action, the same ribozymes can start to interact with these latecomers, meaning that you can have this process of co-option, and co-option is hypothesized to have played a very important role in the origin of the genetic code. So what else can we do with our self-amino acylating in vitro selected ribozymes? Well, some other experiments that we did was encapsulation in vesicles. The first experiments for these ribozymes were done by my colleague Yichen Lai, and it was published in PNAS in 2021. He basically uses a vesicle system form of oleic acid and palmitoyl olein glycerol phosphocholine, POPC for short. So he uses his POPC vesicles, he encapsulates the ribozymes, and then he measures the reactivity with the substrate. And the substrate is semi-permeable, so it doesn't have to be encapsulated when the ribozymes are encapsulated. It can be added later. It's going to go inside the vesicle, and then the ribozymes can react with the substrate, and then we can measure that. And as a control, we have empty vesicles that interact on the external surface with the ribozymes and with the substrate, and we can also measure that activity. So as I said, this was already done, it was published, and we used a novel plausible membrane system that was developed by our collaborator, Dr. Christy Shaw, and she did a vesicle system formed for lipocysteine and laurel-rack glycerol. And this is very interesting because cysteine reacts spontaneously with two short-chain thioesteres to form these diacylipids, generating protocell-like membrane vesicles. So we took this new novel vesicle system, we encapsulated our ribozymes, and we tested the activity inside the vesicle, which is what I'm showing here, and also outside the vesicle. So in the left part of the slide, we have the results for the POP system that Yichen published in 2021. And in the right side of the slide, we have our new results with the novel vesicle system. So as you can see here, inside the vesicle, the Ka, which is the bound fraction or the reactable fraction of the ribozyme, is big, is 61. Whereas our Ka inside the novel vesicle system is very low. So it's a possibility that either the interaction with the new vesicle system and our ribozyme is making the ribozyme not active, or there's another possibility, that is that the substrate is not really permeating through this new vesicle system. So that question can be solved with the outside vesicle experiment. Again, in the left side, you can see the results for the POP system. And as you can see, there's also a very high number for the Ka. And on the right side, you can see that when we don't have the ribozymes inside the vesicle, when they are outside the vesicle or still interacting with the vesicle, the Ki goes up, meaning that the ribozyme is reacting, and meaning that the ribozyme is interacting with the lipids, with the vesicle system, and that interaction is not denaturing the ribosome. So the possible explanation for this first Ka is that there's low membrane permeability of the substrate into the system. But if we go back to the catalytic constants, for instance, the K, which will be the rate of the ribosome, we can see that it's high inside the vesicle. So what this means is that even though the substrate is permeating at a very low rate, once the substrate is inside the vesicle, the ribosome reacts with the substrate. So our ribosomes are actually active inside this novel vesicle system. So we were very happy with those results. And the last example I want to show regarding this in vitro selected self-amino acid latent ribosome is something that wasn't really in the plan when the selection was done, and it's got to do with the chirality problem. So as all of you know, I'm sure, all known life is homochiral, consisting almost exclusively of right-handed sugars and left-handed amino acid. The fact that we have right-handed sugars means that we are going to have right-handed nucleic acids, such as right-handed RNA. So it has been hypothesized that the RNA may have an inherent affinity or an inherent reaction only with L amino acids or their precursors. And this inherent affinity for DNA to L amino acids is what drove the evolution of life. And that's why we have today only the nucleic acids, the RNA with L amino acids and L proteins. But if this is true, this will mean that the RNA word deterministically led to an L protein word. So we realized that our substrate, the amino acid oxazolone biotin, is a very good candidate to study this type of questions, because since we synthesize this in-house, when we have the substrate in solution, we're going to have both chiralities L and D for the substrate represented in equal amounts. So what's happening is that when the ribosomes are reacting with our substrate, the ribosomes are actually interacting with both the L and the D stereoisomers. So if this is true, and there is an intrinsic and inherent affinity for L amino acid from the RNA, then we thought that if we analyze the products of our ribosomes, amino acylated products of the ribosomes, what we're going to find is that the ribosomes only interact with L amino acids, with the L form of our substrate. So we set up an experiment to prove that, and the general methodology goes like this. First, we have to amino acylate the RNA. So the ester bond is formed at the internal 2-hydroxyl to produce the amino acylated RNA. And once we have that, then that ester bond is hydrolyzed, and we get biotinylated phenylalanine. In this case, we use phenylalanine, not tyrosine. And then that phenylalanine is separated from the biotin motif to have only phenylalanine. This phenylalanine is going to be either L or D, and we can measure and identify the chirality of those products by HPLC on a chiral column and then mass spectrometry. So it's a very simple experiment. It takes time, but it's a very simple experiment in which we are analyzing what's the chirality of the preferred substrate for our ribozymes. So what we found is that there's a lot of ribozymes, 9 of the 15 that were tested, that react preferably with the L stereoisomer of the substrate, but there's 6 of them that are actually reacting preferably with the D stereoisomer of the substrate. And this is very interesting because when we analyze the sequence for all of these, all of them are related. And I didn't mention this, but when the ribozymes were discovered, they were clustered into families, and those families are given by sequence similarity. So all of the ribozymes that prefer the D stereoisomer belong to the S1 family, whereas the ribozymes, that interact with the L stereoisomer correspond to the S2 family and to the S3 family. So this is very interesting because let's imagine that in a very early stage of life, we have a variety of ribozymes that are interacting with both L and D amino acids. But if for any reason evolution selected only from the pool of ribozymes that react with the D, or only for the pool of ribozymes that react with L, that could have been the reason why we have the preference for the ribozymes to either L or D amino acids. Either way, we found this. It was very interesting, but one of the questions that originated was that, okay, what if the ribozymes are not actually interactive with the chiral center of the amino acid part of the substrate? It's a huge substrate. It's got biotin. Biotin also have some chiral centers. So what if the ribozymes are actually interacting with some, chiral centers in biotin? So what we did was think, okay, if the ribozymes are interacting with a specific stereoisomer because of the chiral center of that stereoisomer, the amino acid, if we change the chirality of the ribozyme itself, then the preference for the stereoisomer is also going to change. And that's what we did. We made an L ribozyme. In this case was S1A, which, as you can see in this graph here, prefers the D stereoisomer. And when we did the experiment, we discovered that the L RNA reacts with the L amino acid, meaning that when we switch the chirality of the RNA, we also switch the preference for the stereoisomer. And this result is consistent with the main determinants of selectivity being the chirality of the alpha carbon, the amino acid part, rather than other stereoisomer chiral center in biotin. So this means that the chiral preference within RNA system depends on specific structural configurations used for amino acylation, rather than for a general intrinsic preference of dRNA for L amino acids. And also means that the emergence of a world of L proteins was highly stochastic, strongly influenced by the evolutionary trajectories of ribozymes. Now, I have to say here, we're not saying that our ribozymes were the ribozymes present in the early Earth and were the ribozymes that drove the evolution of the genetic code, or were the ribozymes that drove the evolution of the protein translation system. What we're saying is that these phenomena happened in our system, and they serve as a model to understand how the early evolution of those systems, specifically the protein translation system and the genetic code, will have happened. But by no means we're saying that these ribozymes that we selected in the lab were the precursors or were the ones that were, back then. So just as conclusions, I want to say that in vitro evolution, as I mentioned, is a relatively simple but extremely powerful method that allows us to explore sequence space and to discover new functions for molecules such as nucleic acids. It gives us the amazing possibility of reproducing Darwinian evolution in the test tube, and also that the discovery of artificial ribozymes using these techniques has contributed to our understanding of how processes may have occurred in the early evolution of life. And with that, I want to acknowledge the funding sources. As I mentioned, as I'm a NASA postdoctoral fellow, I want to acknowledge the Chen Lab, my PI, Professor Irene Chen, and Dr. Josh Kenchel, which we collaborated together to develop some of these projects, some of these results I just presented. Thank you very much.